morning, friends. I'm Dr. Kiran Ramrao Borkar. Now today I'm going to discuss about carbohydrate metabolism. What is metabolism? The entire spectrum of chemical reaction in living system is called as a metabolism. Before the starting of carbohydrate metabolism, I just explain about the classification of carbohydrate. This carbohydrate classified into four groups, that is monosaccharide, disaccharide, oligosaccharide and polysaccharide. What is monosaccharide? This carbohydrate is made up by single sugar unit. Disaccharide is made up by two sugar units and oligosaccharide is made up by 3 to 10 sugar units. And polysaccharide is made up by more than 10 sugar units. Okay. Now we see the first metabolism of carbohydrate is that the glycolysis. This glycolysis is also called as a EMP pathway. Why it is called as the EMP pathway? Because various step and reaction of glycolytic pathway were worked out by three German biochemists, namely Professor Embedan and Meyerhoff and Parnas. So, this glycolysis is also called as a Embedan, Meyerhoff and Parnas pathway. In short, it is also called as a EMP pathway. Then what is glycolysis? Glycolysis is defined as a stepwise enzymatic breakdown of hexose sugar. And that hexose sugar is that the glucose into two molecules of pyruvate in the cytoplasm. Where? In the cytoplasm. This glycolysis consists of two major phases. First phase is that the preparatory phase, that is starting from glucose to phosphoglyceraldehyde. And second phase is that the payoff phase, that is starting from this glyceraldehyde phosphoglyceraldehyde to the pyruvate. Now we see the first phase of glycolysis is that the preparatory phase and cleavage. And it is the initial phase of glycolysis in which this glucose molecule is activated by phosphorylation and then cleaved into triose phosphate, two molecules of triose phosphate, namely 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. In the next step, only this 3-PGAL, that is the 3-phosphoglyceraldehyde, and therefore this dihydroxyacetone phosphate get converted into the 3-phosphoglyceraldehyde. Thus, at the end of this, two molecules of phosphoglyceraldehyde are formed, include the following reaction. Now we see the first step in glycolysis, and it is the first step in preparatory phase also. That is the phosphorylation 1. In this phosphorylation 1, what happens? This glucose molecule undergoes phosphorylation with the help of ATP. It occurs in presence of enzyme hexokinase and cofactor Mg2+. In this reaction, one phosphate group from ATP is removed to produce glucose 6-phosphate and ADP. The third phosphate group of ATP get attached to the 6 carbon of glucose. It means that here ATP investment occur. Okay. Now we see the second step of preparatory phase and second step of glycolysis also. That is the isomerization. In this step, what happens? This glucose 6-phosphate is converted into its isomer, that is fructose 6-phosphate. It occurs in presence of enzyme phosphoglucose isomerase. Now we see the third phase of preparatory phase and third phase of glycolysis also, that is the phosphorylation 2. During this reaction, fructose 6-phosphate is further phosphorylated by ATP molecule to give fructose 1,6 biphosphate and ADP. This second phosphorylation takes place. Phosphate group is attached to the first carbon. This reaction is catalyzed by the enzyme phosphofructose kinase. This three-step, first, second and this three-step mentioned above the collectively described as a preparatory phase of glycolysis. And the fourth phase is that the cleavage. Fructose 1,6-diphosphate. Fructose 1,6-diphosphate, or it is also called as a biphosphate. Molecule split into two molecules of triose phosphate. This is known as a 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. This reaction is also called as a cleavage, and it occurs in presence of the enzyme aldolase. Now, fifth step of glycolysis or fifth step of preparatory phase is that the isomerization. What happens here? The triose phosphate, namely that is 3-PGAL, it means that 3-phosphoglyceraldehyde, and DAP, that is the dihydroxyacetone phosphor, they are isomer of each other and are interconvertible able. DHAP, that is a dihydroxyacetone phosphate, is converted into the 3-PGAL, that is the two molecules of phosphoglyceraldehyde with the help of enzyme triose phosphate isomerase. Thus, the respiratory substrate is now in the form of two molecules of 3-PGAL. It means that two molecules of 3-phosphoglyceraldehyde. Now we see the second step of glycolysis, that is payoff phase and oxidative phase, that is starting from 3-phosphoglyceraldehyde to the pyruvate. Now we see the sixth step of glycolysis and first step of the payoff phase, that is phosphorylation. What happens here? In presence of enzyme phosphoglyceraldehyde dehydrogenase. This 3-phosphoglyceraldehyde undergoes dehydration. It means that oxidation. Simultaneously, it undergoes the phosphorylation. That is, inorganic phosphate get incorporated to give 1,3-bisphosphoglycerate, or it is also called as a 1,3-diphosphoglyceric acid. During this reaction, coenzyme NAD take up 2 hydrogen and get reduced to the NADH2. Now we see the seventh step of glycolysis and second step of payoff phase is that the ATP generation 1. Okay. In this step, what happens? 1,3-diphosphoglyceric acid or 1,3-bisphosphoglycerate is converted into 3-phosphoglyceric acid. It is also called as a 3-phosphoglycerate by removal of phosphate group to produce an ATP molecules. The reaction takes place in presence of enzyme phosphoglycerate kinase. Now we see the 8th step of glycolysis and 3rd step of payoff phage, is that the isomerization. The enzyme phosphoglycerate mutage brings about the conversion of 3-phosphoglycerate into its isomer 2-phosphoglycerate. In this reaction, phosphate group is shifted from the 3rd carbon atom to the 2nd carbon atom. Okay. Now we see the 9th step of glycolysis and 4th step of the payoff phase, is that the dehydration. What happens here? This 2-PGA, that is 2-phosphoglycerate, undergoes dehydration. It means that 1 molecules of water are removed from this 2-phosphoglycerate. And phosphoenol pyruvic acid, in short it is also called as a PEPA, is formed. The enzyme enolase catalyzes the reaction in presence of Mg2 plus as a cofactor, and 1 molecule of water is released. Now we see the 10th step of glycolysis and 1st step of the payoff phase, is that the ATP generation 2. On hydrolysis, phosphoenol pyruvic acid loses a phosphate group, that is dephosphorylation, and gives pyruvic acid and pyruvate. The released phosphate group combines with ADP and ATP generation takes place in presence of enzyme pyruvate kinase. One glucose molecule is converted into the 2 molecules of pyruvate at the end of the glycolysis, as each reaction in the oxidative and payoff phase from 6th step to the 10 above occur twice. Therefore, 2 ATP molecules, that is 2 ATP molecules are generated at the step 7, and 2 more ATP molecules at the step 10. In all, it produces 4 molecules of ATP, 2 molecules of NADH2 are produced at 6th step. The overall equation for glycolysis may be written as glucose, that is 6 carbon, plus 2 NAD cofactor plus 2 ADP plus 2 inorganic phosphate. It gives rise to 2 pyruvate, that is 3 carbon, plus 2 NADH2 plus 2 ATP. Okay, now 2 ATP molecules generate here. Thank you so much.